Daniela Comelli from Politecnico di Milano. And just before starting the presentation, a few words about my professional location that is in Milano, at the Politecnico di Milano, and specifically at the physics department of Politecnico di Milano, where in terms of research activities, there are 5 research lines. And the one I'm involved in is the third one that is called Photonics for Health, Food and Cultural Heritage. That means that we develop and use photonics technologies for a variety of applications. We do a lot of applied research. My main expertise in the application for cultural heritage. And in this sense, I collaborated in the past and I hope also in the future with people from FORF. So, we are also a node of laser lab infrastructure, and specifically we are referred to as CUSBO, Center for Ultrafast Science and Biomedical Optics. So in case you are interested in using our time-resolved facilities and super fast laser sources, you can apply as a part of laser lab. So let's go to the real part of the presentation that deals with fluorescence. So here I just start with a slide that usually is put at the end of the presentation, but it's just to let you appreciate the manifold use of fluorescence imaging studies for very different applications. So I start giving you some ideas of what are the advantages of using fluorescence imaging as spectroscopy with respect to other spectroscopy techniques, or better in combination with, because we know that multimodal application now it's really a benefit. So fluorescence has different advantages. First of all, it has a high specificity, especially when it is used in combination with a fluorescent marker or tag, because we can, for example, tag different parts of a molecule, of a cell, and so localize them thanks to fluorescence. Or we can use fluorescent probe that can grow together with some specific cells in a mice, and so see the evolution of these cells that in this case are tumor cells. So it's a very specific technique, but further it is an optical technique, and thanks to it, it's a fast technique. With it, we can track very fast phenomena. For example, here we can do live imaging of mitochondria. Further, it has a high sensitivity in the sense that we can detect a trace amount of a fluorescent compound if the other compounds present in the sample are not fluorescent. For example, this is an application in the cultural heritage field, where we look at the near-infrared emission of a specific pigment that is known as Egyptian blue. As you can see, let me just put my pointer visible, okay. Here you can see it's a luminescence emission in the near-infrared that is related to the blue parts in this archaeological finding. But actually, this is clear because they are blue in color. But we can see here in these archaeological remains, as it is a mask, that even in the area that invisible are not apparently blue, we can recognize its presence thanks to the presence of this luminescence pigment, thanks to its emission. So it's a very high sensitivity technique. And last but not least, it can be used in the laboratory, but it also very portable. This is, again, an application on cultural heritage for the study of Michelangelo's David, marble surface, but There are many applications also in medicine and directly in cleaning during surgery through endoscope to assess the fluorescence during a clinical operation. So these are some of the advantages of fluorescence. In order to understand all these advantages, it is important to know the technique. So in my presentation, I will give you an introduction to fluorescence properties, a focus on time-resolved fluorescence imaging, also referred to as fluorescence lifetime imaging, a few words on the many different fluorescence probes, and then I have tried to find out in the literature some recent application that could be helpful considering the aim of these projects, of the remade projects. Okay, so let's go to an introduction to fluorescence. And for describing fluorescence, it is necessary to consider a molecule and to consider the energy levels of a molecule in terms of electronic level and vibrational levels. So these energy levels that are related to the orbital motion of electrons and to the relative motion of atoms within molecules, so to the vibration of the molecule. This is a picture of a possible Jablonski diagram where electronic energy levels are depicted as bold, and within each electronic energy level, we can see the different vibrational levels related to it. And then let's see what happens if we try to excite a material by some extra energy that can be, for example, provided by light. And in this case, we speak of optically induced fluorescence or photoluminescence. And so what happens is that the material is excited to an excited state where typically to a certain vibrational level of this excited electronic state. And from this, there is very quickly a vibrational, the excitation to the lowest vibrational level of this excited energy. And then from this excited state, fluorescence can occur. And this happens if the molecule de-excites to the ground state via emission of light. The phenomenon is referred to as fluorescence. Actually, other phenomena can happen because once the molecule is in its excited state, it can also, in some cases, give rise to an intersystem crossing, and so going to another triplet, an excited state, which is typically the transition from this energy level to the ground state is typically not permitted, or better, it can happen, but with a much lower probability. And if this happens, we call the emission of light corresponding to this excess energy phosphorescence. So this is a brief and a schematic picture of the phenomenon of fluorescence and phosphorescence. Both of them are generally referred to as luminescence. That is the more general term for them. While fluorescence is the emission from a permitted transition, and phosphorescence is the emission of light from a non-permitted transition. Okay, so when we look at fluorescent compounds, the first aspect that we note is the color of the emission, and this is a picture that let us appreciate different fluorescent dyes, each of them with a different color of the emission, that from a physical point of view, this color of the emission is related to a property that is the emission spectrum. That simply is the distribution in energy of the emitted light, while the absorption spectrum is the distribution in energy of the absorbed light. So how we can relate the emission spectrum with the distribution in energy, with the Jablonski diagram? So let's go back to this picture that is not so different from the Jablonski diagram, we are forced to remind a principle that is called the Frank-Condon principle, that say that electronic transition, that means absorption or emission, are essentially instantaneous if compared to the time scale of nuclear motion. And with this principle, we can depict this absorption and emission transition as vertical lines. So what happens when we excite a molecule is that from its ground state, we have that the material can populate the different vibrational levels of the excited state. So the absorption spectrum that is not now typically is not a line as it happens in gases, but when you consider liquids or solids, you have that the absorption spectrum is broad. It is broad because when exciting the material, the absorbed light can put the molecule in all these vibrational level of the excited electronic state, while following internal conversion, once the material has been excited, the molecules very rapidly fall in the lowest vibrational level of the excited state, and only from this state, fluorescence can happen, emission can happen, and thus the emission spectrum resembles the distribution in energy of the vibrational level of the ground state, because from this state, the emission can go down, can put the molecule into the many different vibrational levels of the ground state. And this is the reason why the emission spectrum is broad, with a specific pattern. Sometimes it is possible to see this specific pattern where each peak is related to a transition from a specific vibrational level of the ground state. And typically, actually not typically, it can happen that if the distribution of the vibrational levels of the excited state and of the ground state are similar, then the absorption and emission spectrum are mirrored. This is the so-called mirror law. And this is an example for a dye that is called body P, where you can see that effectively the excitation and emission spectrum are symmetric in terms of with respect to wavelength. But for many different reasons, we have no time for explaining this specific stuff. There are many exceptions to this. You also here provide you some example of emission spectrum that do not respect this rule. But despite this, it is important to remember this interpretation of the emission spectrum of a compound. But apart from the emission spectrum, there are other important properties of luminescence that has to be taken into account. The first one is the so-called quantum yield of fluorescence, or more in general of luminescence, that is simply the efficiency of the emission process. It has the ratio between the number of emitted photons and the number of absorbed photons. So let's have another scheme that allows us to better describe this parameter, the quantum yield. So imagine this is a simple view, again, of the energy levels of a molecule with a ground state and the electronic excited state and the force vibrational levels from which the excitation can happen. So imagine that we have excited a molecule, and once it is excited, the excitation can happen via emission of light with a certain rate, with a certain probability. And the rate means the probability for this process to happen. But actually, it is typically very normal that the excited population can relax back to its equilibrium state, also via non-radiative paths, with a rate that is referred to the non-radiative de-excitation rate. So these are, in general, the two main paths for the excitation of the molecule from its excited state. And considering these two rates, it is possible to understand the quantum yield is the ratio between the rate of radiative decay process with respect to the sum of all the possible rate of all the possible excitation paths. And obviously it is a number that is minor than 1. And this is the definition of the quantum yield. That is a very nice parameter to be measured, but it is not so easy to measure it, because for measuring the quantum yield of a fluorophore, we need to do absolute intensity measurements that are never easy to be performed. So we can consider also the emission lifetime. It is defined as the average time a molecule lasts in its excited state before returning to the ground state. And for both fluorescence and phosphorescence, we can say that they follow fourth-order decay kinetic. That means that the population decay kinetic is proportional to the population amount in the excited state. And thence, by integrating this differential equation, we can understand that the population evolves in time as an exponential decay, whose lifetime is exactly the inverse of the sum of the non-radiative decay rate and the radiative decay rate. And thence, if a luminescence emission, when is excited by a pulse of light, decay in time as an exponential decay. And it's interesting to note that both the quantum yield and the lifetime are sensitive to the same parameters, the radiative and non-radiative decay rate. And so it is possible to get the same information of the quantum yield by measuring the lifetime. And the measure of the lifetime is easier than the measure of the quantum yield. And indeed, one of the most interesting features of the emission lifetime is exactly the fact that it depends on the non-radiative decay rates, because it means, thanks to it, that it is a parameter, that it is a phenomenon, a parameter that is sensitive to the fluorophore microenvironment, because for any time the fluorophore, for some reason, has a microenvironment that modifies the non-radiative decay rate, the lifetime can probe this change in the microenvironment. And this is one of the most effective properties of fluorescence lifetime. So here I just remember you some order of magnitude. Fluorescence time typically range from hundreds of picoseconds to tens of nanoseconds, while the lifetime of phosphorescence ranges from microseconds to milliseconds, in some cases even seconds. So this is just to stress this aspect, that is the sensitivity of the lifetime to the microenvironment, so to microenvironment parameters as the viscosity of the solvent, ionic strength, concentration of ionic species, binding sites. And this is an example of this property, because you can see a map of the fluorescence lifetime of a dye, a fluorescent dye that is known as DASPI, in ethanol mixed with glycerol, with increasing in this direction, with increasing concentration of the glycerol for increasing the viscosity. And you see that while the viscosity of the solvent increases, the lifetime decreases, because we add the non-radiative, we increase the non-radiative decay rate of the fluorophore. And so we can probe the viscosity of the solvent thanks to lifetime. Another possibility is that we can probe the pH of the microenvironment. And this is a very, very interesting application for studying tumors and understanding the presence of tumors in cells, because tumoral cells can change the pH of the microenvironment. And so here we have an example of a cellular tumoral cells characterized by different pH value that display a fluorescent emission, but more interesting, if we look at the lifetime map, we see that at variable pH, we detect different lifetime values. And so again, we can probe the pH of our microenvironment. Another very interesting properties of fluorescence lifetime, of in general fluorescence, is a phenomenon called FRET, that means Forster resonance energy transfer. That is very useful because it can be used to probe the distance, the nanometric distance between two compounds, between two fluorophores. So let's imagine to have two fluorescent species. One is defined as the donor, and the other one is defined as the acceptor. And what can happen is that following excitation of the donor, if the emission spectrum of the donor overlaps with the absorption spectrum of the acceptor, there can be a non-radiative transfer of energy from the donor to the acceptor. And this, the interesting thing is that it can happen, this non-radiative energy transfer, only if the two molecules are very close to each other. Very close means at the distance of few nanometers. These are the conditions that allows for this phenomenon to take place, that I have already synthesized. And if, so these are two pictures that display what happens if the two fluorophores are too distant. If I excite the donor, then the donor just emits its light. But if we put the two fluorophores in close proximity by exciting the donor, what happens is this energy transfer, and instead of having the emission of the donor, we have the emission of the acceptor. And the efficiency of this phenomenon is directly related with the distance R between the two molecules. And so we can probe this phenomenon by looking at the intensity of the emission of the acceptor. This is one possibility, because I excite the donor can emit if the molecules are too distant, but if they go in close proximity, I will get the emission from the acceptor. But although this is feasible, and actually it is done, this method suffers from the fact that it is susceptible to variation in fluorophore concentration. Again, I have to make a measurement that is absolute in intensity. I can also measure this parameter, sensitive to FRET, by measuring the lifetime, because you can consider that when the donor transfers its energy to the acceptor, again, we depopulate its excited population via a non-radiative decay rate. And so we can imagine that the lifetime of the donor will decrease in lifetime, in value. And so we can probe the efficiency of the FRET phenomena by looking at the lifetime of the donor. And the lower is the lifetime of the donor, the higher is the efficiency of the FRET phenomena. And this is an example of this application with two different fluorescent dye that when they are not close proximity, we can look at the lifetime of green fluorescent protein, and when they get in close proximity, we observe a net decrease in lifetime. And so fluorescence lifetime imaging is super useful for sensing FRET phenomena. Okay, so here is a basic of fluorescence properties. Now let's see how we can perform. I have let you appreciate how fluorescence lifetime imaging is useful, but we have to understand how to realize a setup of fluorescence lifetime imaging. So, first of all, we have to remember that this is an imaging technique. So, the question is how to create the image of our specimen. And typically there are two ways. One is to use a detector that is a wide field detector. So it is an area sensor that collects the fluorescence from a sample area, or by a spatial scanning approach. So we focus light on a point, and then we do, with a raster scanning, point-by-point scanning, we analyze the whole sample. Both the approaches are widely used. I start with the second one, the spatial scanning approach. That means to excite point by point each point of a sample with a pulse picosecond laser excitation. Typically, the excitation is a repetitive pulse excitation with a high repetition in the order of megahertz. And then once the fluorescence of the material is excited, we have to collect it with a fast detector with picosecond temporal resolution. And typically this is done by using a method of detection that is called time-correlated single photon counting detection scheme. Here there is a very super schematic representation that means to excite our material with repetitive pulses and collect the fluorescence that here is schematized just as a pulse, but actually lasts for some time, by measuring for each pulse the delay between the excitation and the collection of the event of the emission. And doing this for each point, we can reconstruct the emission decay and then reconstruct the lifetime. What does I explain it a little bit better? So imagine to have a pulse of light, and when the material is excited by this pulse, we collect just one single photon. That means this is the reason why I call it time-correlated single photon counting. And we measure the delay, the time that occurs between this starting event and this collection event, that is the stop. If we repeat the measurement many times, we create a kind of probability, an histogram of start-stop times, and then at the end we reconstruct the transient decay kinetic of the emission. This is the electronics behind this setup. I don't go too much deeply in this, but it's a very sensitive techniques, because we are sensitive to single photon event, that allows us to record fast phenomena with picosecond temporal resolution. We need detectors that are able to have this fast response, and typically 3 main types of detector can be used for time-correlated single photon counting. They can be photomultiplier tube. They are, let's say, the older detector used in this type of collection scheme. The advantages are that they are rather inexpensive. They provide a high amplification gain for the single photon detector, because here you have this photocathode that allow the conversion of the single photons into an electrons, and then the amplification provided by the diodes, and then we measure the currents created by all these photoelectrons. The limits of photomultiplier tube is that they are characterized by a temporal response of the order of nanosecond. Better than photomultiplier tubes are microchannel plates that are rather similar. Again, we detect one photon that is converted into photoelectrons and then multiplied by the microchannel plate. The advantages is that it has a higher temporal resolution. They are a little bit expensive and fragile. Another more recent use detector is the avalanche photodiode, and also the single photon avalanche photodiodes known as SPAD, that are the semiconductor version of a PMT. They are a very fast response, let's say the best in terms of temporal resolution among these 3 type of detector. they have a lower internal amplification factor, and the main limit is actually the area that is rather small. Whichever is the detector that you use, what happens is that at the end, you have created a setup that point by point can reconstruct the transient decay kinetic of the emission, and starting from this transient decay kinetic, you can reconstruct the lifetime map, and here you see this beautiful image of the cells, see in intensity, and then convert in the lifetime to provide a wealth of information. And typically there are many commercial devices based on this method that allows for sample to be studied under the microscope, even with 3D reconstruction, so complemented with a confocal or a two-photon microscope. The advantages of this method is that we have really a very good temporal solution with picosecond or sub-picosecond temporal solution. As I told you, they can be coupled to an optical microscope. It can be easily implemented with multiple excitation wavelengths and multiple collection in multiple spectral bands to add in some way the information about the core of the emission. The main limit is that it is typically confined to the laboratory. So by coupling this detection scheme to a microscope. On the opposite, we have the wide field imaging approach that uses a large sensor, excite the sample in its whole sample surface with a larger laser beam, and then we need to collect the fluorescence with a fast imaging camera. I start with the advantages, or actually the features of this approach with respect to the previous one. In terms of temporal resolution, typically this kind of approach gives rise to a lower temporal resolution, generally starting from the nanosecond temporal resolution. On the other hand, again, this approach can be implemented with multiple excitation wavelengths and collection in multiple spectral bands. The main advantage, it can be coupled with different optical collection setup. So microscopy, but also mesoscopy, macro scale, large field of view, and indeed it can be easily implemented as a portable setup. And for example, here this is an example of using it for analysis of large mural paintings, but as I told you, also in clinics. For sure, this method is more versatile, more flexible than the previous one. For what concerns the wide field imaging approach, there are two main approaches. One is the direct one, the time domain approach, and the other one is the frequency domain approach. I briefly presented the frequency domain approach. That means to excite the sample with a light provided by a laser on a LED that is modulated in frequency, with a modulation that typically is in the megahertz order of magnitude. And following this excitation, the emission is also modulated. But what happens is that the emission is shifted, is phase shifted with respect to the excitation, and this phase shift is exactly related with the lifetime of the emission. So if we are able to measure this phase shift, we can reconstruct the lifetime. So this is what I've just said, and this is the formula, actually the definition of the phase delay. You note that the emission suffer also for a demodulation ratio, and this is the basic scheme for performing measurement in the frequency domain approach with the homogeneous detection scheme that use a modulated laser source, but also a sensor, typically an image intensifier that whose gain is modulated with the same frequency. And with this homogeneous scheme, we can probe the shift in phase of the fluorescence signal by probing it with a 2 pi and then reconstruct the lifetime. It is just interesting to note a very a rather recent approach for analysis of lifetime with the frequency domain approach. That is, once the lifetime signal from in the so-called phasor space. That means that once the lifetime signal from every pixel is measured, you can Fourier transform into frequency components, a phasor, that can be charted on a phasor plot. And it is interesting to note that in this phasor plot, this circle is related to mono-exponential fluorophore, to mono-exponential components, but actually often our compounds are characterized by a multiplicity of lifetimes. And so we can see how they can be correlated with the movement from one component to another one component without any need for a real mathematical modeling. And this is, for example, an application to NADH, where you can see the transition in lifetime between unbound NADH and bound NADH, and this is the measured population. So we can try to provide an interpretation of our data, even visually, in this way, okay? But there is another way that is the direct one. So we excite our fluorescence with the laser pulse, and then we looked at the decay of the emission, and this is called directly lifetime-based imaging. For doing it, we need, to temporarily sample our fluorescence signal with a fast detector that collects all the photons falling emitted at a certain delay with respect to the excitation pulse. For doing it, we typically use a time-gated image intensifier that is a kind of image camera characterized by a very fast electronic shutter. So it is made of a photocathode, a microchannel plate, and then phosphor, where the light is converted into photocathode, but when the photocathode is kept at a positive voltage, photoelectron generated just remain at the photocathode. But if we then apply a high negative voltage at the photocathode, the electrons are sent to the microchannel plate, amplified, and then, thanks to a further positive voltage, are converted to phosphor, and here we get light amplified. So here we can see the intensifier in its off state and its on state. And the nice thing is that we can provide this on state in very narrow temporal window as short as hundreds of picoseconds. And so we have created an image camera characterized by a sub-nanosecond electronic shutter. And for doing this, we just measure the excited fluorescence and collect sequential images of the fluorescence at different delays with respect to the excitation. And with the sequence of images, we can then apply some mathematics and reconstruct the map of the lifetime. So this is fluorescence lifetime imaging in the wide field approach. Just briefly, because I can skip this part because it's just mathematics, so that the real thing is to tell you is that if I have one single fluorophore, I would have a mono-exponential decay. But telling the truth, in real sample, this rarely happens. And so what happens is that we can have two populations, or more generally, a continuous distribution of lifetimes that can be fitted, for example, with a model that is called the stretched exponential function. But there is in the literature really a lot of mathematics for fitting multiple exponential decay. So I will go again rather briefly in this section because just related to tell you that why we can use fluorescence, because there are many molecules that are fluorescent. And in fact, we can have natural molecules that are fluorescent. We call them endogenous fluorophores. For example, amino acid and proteins, collagen and elastin. So generally, all organic stuff if excited when UV light is fluorescent. But the limit in using the fluorescence emitted by natural molecules is that they can be rather specific. So in many applications, it is more convenient to use synthetic fluorescent probe, exogenous fluorescent probe, as synthetic colorants, as this one, that can be precisely tagged to a molecule or to a specific binding site to achieve this very nice view of for example, mouse intestine with its different part tagged with a different fluorophore, but also genetically encoded fluorescent probe that can be really used for understand a lot of stuff in biology. But what I think is more interesting for you is to understand potential application for, I imagine, related the remade project. So I try to find something in the literature, and I hope to have get the point or attract your curiosity. So I look at potential use of fluorescence lifetime imaging for the detection of microplastics in our environments, and I found that there are some attempts for detecting a particular type of plastic in the environment thanks to its natural fluorescence. And there you can see in this lifetime map how this HDPE plastic is different from the soil and the spruce surrounding it, but probably more effectively by targeting different type of plastic with a specific fluorophores, in this case, a dye called Nile red, whose lifetime is changed when, depending on the type of plastic it is tagged on. And so here are lifetime maps where different types of plastics are characterized by a different fluorescence for the emission of Nile red. And so it is very useful for discriminating between different types of plastics. Then another potential application I was thinking was for the creation of device like solar cell. Actually, they are called luminescent solar concentration that are this type of device made of a sun irradiation collector. And in this slab, there are many different types specifically built, specifically created of fluorophores that can absorb the radiation emitted by the sun, emit light that then is waveguide through the photovoltaic cell. And they are so a kind of photovoltaic application for very large area where you need to have transparency. And there is a lot of study for creating, for encoding specific fluorescent probe, typically quantum dots, with specific emission and absorption spectrum, so to give rise to very efficient luminescent solar concentrator. Then I was thinking I had a very well-known application of perovskite solar cells, where fluorescence, luminescent perovskite are generally photoluminescent, and time result study of photoluminescence is useful for measuring some specific parameter of perovskite related to the carrier mobility and recombination rates. So to design the best perovskite material with improved electroluminescence quantum efficiency. And this is enough. This is for those among you that want to deepen their knowledge on fluorescence imaging and fluorescence lifetime imaging. I think this is a very nice review of the most recent application and use of fluorescence lifetime imaging. And with this, I think I finished with a little bit delay, but that's enough. This is just a picture of my group in Politecnico. So thank you for your attention, and I'm open to any curiosity question, even far from this presentation.